Okay. So we're talking now about biological membranes and transport systems. Yan, Dr. Aksay. If you have any questions, you can message in chat or you're free to unmute yourself and ask your question here during our pauses where I ask when when I allow for asking questions. Okay. So biological membranes. First, learning objectives. We want to discuss all three basic components essential to the composition of your biological membranes. That you guys are able to compare and contrast between phosphoglycerides, sphingolipids, and sterols. Uh, Also, differentiate between integral and peripheral membrane proteins. Then we should be able to correlate the composition of the biological membranes to the properties in the formations of micelles, vesicles, and the lipid bilayer. And in your own words, be able to explain your understanding of the fluid mosaic model. Then we should be able to differentiate the different forms of molecular transport systems and then familiarize with the types of membrane receptors. We won't go too deep on the membrane receptors type. I would let physiology do that for me. Okay. So first and foremost, we're talking about biological membranes. Best way I want to start is I want you to think of it as something like the skin to a human, where that where you sorry, where it is akin to the skin of a human. Okay. So what is human skin for us? It is a mobile and dynamic organ that consists of proteins and connective tissue. The skin and the external parts of the body help compartmentalize the regions of the body. Kung epidermis, dermis, connective tissue, and below. The skin is selectively permeable. Pag nababasa ka, may pumapasok do the cells accept water or not? Does it reject certain things or not? The skin separates the inside of the body from the outside. The skin is able to perform specialized functions, particularly certain parts of the skin, like the skin found on the lips, on the genital organs, and among other areas. And of course, it also allowed sites of interaction with the environment and among other individuals. In terms of biological membranes, kind of like the same thing. Okay? Biological membranes are dynamic and highly fluid structures, where in this case, it consists of a lipid bilayer with proteins scattered all throughout. They define cell boundaries and act as a barrier. They separate one cell from another, and they can block the entry of other molecules going in and coming out. Biological membranes have selective permeability. It allows certain things in and out and rejects certain things from going in or out. It maintains differences in composition between the inside and the outside of the cell. It has, instead of special organs, contains proteins that perform specialized functions. And then they have also sites of interaction with the environment and among cell-to-cell contact. Okay. So as we discuss the biological membrane, let's first start with the components. There are three major components akin to the three major macromolecules that we have been discussing since the start of the year. We have your lipids, your proteins, and your sugars. When it comes to your biological membranes, lipids take uh, the most in terms of molecules and in mass. Second are your proteins, and then your sugars. So let's start first in lipids. You have three types, sorry. Your phosphoglycerides, your sphingolipids, and your sterols. So let's discuss that. Phosphoglycerides. As the name implies, it's a phosphorylated glyceride, which consists of first the glycerol backbone you see here in green. Glycerol being a 3-carbon structure with a phosphate head esterified to C3. Esterified meaning it contains an ester bond, which is in this case the carbon 3 of your glycerol backbone connected to a single bond with oxygen, bridging to a phosphate PO3. Okay. And then for carbons 1 and 2 of your glycerol backbone, you have 2 fatty acid tails that are also esterified. So you have one long hydrocarbon here and a second long hydrocarbon here. Phosphoglycerides, what you can see here is the base form of the phosphoglyceride. There can be hats, quote unquote. I call them hats because it attaches to the phosphate head that can attach into phosphate. Examples. First, the fatty acid tails that are connected to your phosphoglycerides are typically even numbered carbon molecules. So they're molecules with even numbers of carbon, commonly around 16 to 18. Typically, they are all unbranched, and then they are either saturated or unsaturated. Typically, the fatty acid attached to C1 or carbon 1 is saturated, and the one attached to carbon 2 is typically unsaturated. So you see here in table 12.1, this is found in Devlin. Devlin is my major reference, although we also use Harper. So we have these common names and the systemic names of the other fatty acids found in your glycerophospholipids. Going back to the phosphate hats. Your phosphatidyl is your base molecule that can have hats such as choline, ethanolamine, serine, glycerol, and inositol. These will also form an ester bonds with the phosphate head of your phosphoglycerides. When you combine them with your phosphatidic acid, you will have phosphatidyl choline, phosphatidyl ethanolamine, phosphatidyl serine, phosphatidyl glycerol, and phosphatidyl inositol. These are different forms of phosphoglycerides that can comprise your biologic membrane. Next, we look at sphingolipids. This time it consists of a sphingosine backbone. Sphingosine is here in green, where it first has a 3-carbon chain with 2 hydroxyl groups, one in your alpha carbon 1 and one on your carbon 3, with an amine group, NH2 amine at C2, and then a long hydrocarbon chain attached at C3. So here is your base sphingosine, and then here is dihydrosphingosine, where the difference is there is no double bond between carbon 4 and carbon 5. What's more important here is it has the amine at C2 and a long carbon chain at C3. So that is your sphingosine backbone. Of course, to become a sphingolipid, a fatty acid chain will be chained via amide bond. Your NH2 will become NH, connecting to a carbon bond to form a ceramide. Typically, what attaches here also to your sphingolipid is unsaturated fatty acid. So if you notice again, you have your sphingosine head, and similarly, two long hydrocarbon tails. Much like the phosphate group of uh, phosphoglycerides, you can have groups that connect to the hydroxyl group of C1. So this can have other hats as well, like phosphocholine, when connecting to your ceramide, now forms sphingomyelin. Apart from phosphocholine, it can be also phosphoethanolamine. So sphingomyelins are, in a sense, ceramides with phosphate group hats. So combined with phosphoglycerides and sphingomyelins, because they also contain your a phosphate hat, they are what you call your phospholipids. So here it is, your phosphoglycerides and your sphingomyelin will be your phospholipids. So for each cell membrane, they have distinct fatty acyl groups. So certain parts of distinct cell regions can have distinct fatty acyl groups. Okay, so certain parts of the cell may have higher amounts of stearic acid. Some will have more oleic acid. This determines their function and the properties that they have. Okay. Their existence within the cell also differs. So as we see here in this table, glycerophospholipids are more evident within the inner mitochondria. This is glycerophospholipids are in blue. More prominent in the inter and the outer mitochondria are about less than 50% in the plasma, where sphingolipids have a greater concentration. In the inner mitochondria, we see trace to no amounts of cholesterol, while other forms of lipids can exist in the plasma and the Golgi apparatus. At the same time, among phospholipids, their forms can exist in different concentrations. In red, you see phosphatidylcholine greatest within the rough endoplasmic reticulum and the nucleus, as compared to the Golgi apparatus and within the plasma. Within the plasma, you will see greater amounts of phosphatidylethanolamine, also within the mitochondria. So their concentrations differ based on the locations of the cell have different concentrations of lipids and the different compositions of lipids in their concentration. Okay. So typically your phospholipids can have saturated fatty acids, and with trans unsaturated fatty acids, they present as straight chains. You will see here, this is a saturated fatty acid with no double bonds. The typical angle of the typical angle that they form is about 110 degrees. No need to be specific on these numbers. These are just examples that I want to show. But particularly when you have double bonds, cis double bonds, you form kinks within the hydrocarbon chain because those do not fold into a pattern that is relatively straight. They form a tail or they form a kink in a direction not parallel to the rest of the hydrocarbon chain. Why is this important? I will explain later. Next, with the sphingolipids is glycosphingolipids. So glyco meaning is sugar. This time you have a ceramide or a sphingoside with a attached fatty acid now links to a sugar via beta glycosidic bond to your C1 hydroxyl group. Examples of these are cerebrosides, where the sugar is either glucose to form glucocerebroside or galactose, which forms galactocerebroside. Notice being glucose and galactose, they are monomeric sugars as compared to gangliosides, which uses oligosaccharides. Yeah. Okay. So cerebrosides have monosaccharides, gangliosides have oligosaccharides. And for gangliosides, their oligosaccharides can have one or more residues of sialic acid. So here's a picture that shows your cerebroside. You have your sphingosine backbone. You have your R group attached to your amine, your fatty acid attached to your amine, your long carbon chain attached to carbon 3, and then esterified to your hydroxyl group of carbon 1 is this time a single sugar residue. That is a cerebroside. Your ganglioside this time is esterified to in this case, a 4 uh, 4 sugar moiety of uh, oligosaccharide that contains one branch of sialic acid. So that is a ganglioside. Third, we shall check on sterols. The most common sterol in membranes for animals is cholesterol. As we see here, cholesterol is a compact, rigid 27 carbon structure, contains a 8 carbon tail attached to carbon 17, and more importantly, a polar hydroxyl group at C3. These cholesterols can be found within the plasma membrane, usually in between your other lipids. They act as a rigid structure and they modulate fluidity in response to temperature changes. We'll discuss that um, as we discuss fluid dynamics. So again, your three forms of um, lipids is typically in your phosphoglycerides, your sphingolipids, and then your cholesterol. Next, we talk about proteins. The proteins provide structure to the cell, and they can act as receptors for bindings of hormones. They can be facilitators of transport for molecules in and out of the cell, and they can be enzymes that are involved in transduction. So your proteins are your more reactive groups, while your lipids are your main structural components. These are examples of the functions of plasma proteins. They can exist as channels, enzymes, receptors, structural components like attachment to the cytoskeleton, and cell adhesion, or provide identity markers. When we talk about membrane proteins, we first have to classify them as either integral or intrinsic or peripheral. So when we're talking integral proteins, these or intrinsic proteins, it's in the name. They are integrated into the lipid bilayer. They may also traverse the layer once or multiple times. In order to remove intrinsic proteins, you have to, in a sense, destroy or cleave a certain area of the of the bilayer. You cannot remove them without disrupting the bilayer. They are connected to the bilayer through a series of non-polar amino acids, which we've discussed. Those are your aliphatics and your aromatics that attach with hydrophobic tails. And then, of course, as it has two hydrophilic ends with a membrane spanning hydrophobic region. So I will enlighten that little later when we discuss the bilayer. Your peripheral proteins, conversely, are loosely attached to the surface, either in the top or in the outside surface or in the inside surface. They are predominantly bound to hydrophilic regions rather than hydrophobic regions, and they may not always be attached to the lipid bilayer. They can be potentially transient. Here's a diagram that shows the difference between integral membrane proteins, where you have a protein that spans the entire membrane. Will have hydrophilic ends in the top, in the extracellular, in the cytoplasmic side, and an internal area that is composed of hydrophobic amino acids. They can span once or multiple times. As compared to peripheral membrane proteins, where they do not span the entire surface. They may be linked primarily to the hydrophilic layers instead, or not completely throughout the membrane. Okay? Understood naman yung difference between integral and peripheral membrane proteins. Any questions so far? No questions? Okay. You want a copy of the PowerPoint now or after the lecture ba? Would you prefer?
Doc, if possible daw po kaya puwede daw po kaya ngayon para makasunod po sa lecture.
Okay, let me just save the PowerPoint because medyo malaki siya.
Thank you po, Doc.
Okay. Stop share muna ako. Uploading na.
Thank you po, Doc.
Okay, habang nag-upload, mabilis lang. For membrane carbohydrates, they're present usually as oligosaccharides simply attached to your proteins, hence they become glycoproteins or glycolipids. So they don't exist without any other attachments. Otherwise, they will become free sugars. Okay, so you will typically see your carbohydrates on the external surfaces, where the glycoproteins and the glycolipids form what we call a glycocalyx. The role of the carbohydrates then are more on cell-to-cell recognition and adhesion, and occasionally with receptor action. Okay. Ayan, na-upload na. Okay. So now that we know the three main components of your membranes, which is your lipids, your proteins, and your carbohydrates. Among your lipids is your phospholipids, your sphingolipids, and your cholesterol. Your proteins naman are multiple types of proteins but classified into your integral and your peripheral proteins. The carbohydrates, again, they are always attached to a base protein or a base lipid to form glycoproteins or glycolipids. So now that we've discussed the contents of your membranes, now let's discuss its properties. There are 6 main properties that we want to go through. That the properties of the components and the layer, the membrane in itself. So first, let's talk about amphiphaticity or amphiphatic. The property of being amphiphatic is the property of a molecule that containing both a hydrophilic end and a hydrophobic end. So you keep seeing these structures of one polar head with two non-polar hydrophobic chains. It's typically what you are going to see in diagrams of a lipid bilayer. Okay, so the head is your polar group. The two long hydrocarbon chains are your non-polar group. If you take the diagram of your phosphoglyceride and phospholipid, sorry, and your ceramide or sphingolipid, they will produce generally the same shape and formation. So your two hydrophobic tails attached to carbon 1 and carbon 2, plus your polar head attached to carbon 3. Hence, it looks similar to the structure of a head with two tails. The same with your ceramide or your sphingolipid, where you have a polar head with two non-polar tails. Okay, again, all three lipids have your amphiphatic senses. Your phosphoglyceride, your sphingolipid, and even your cholesterol. Cholesterol has a small polar area that contains a hydroxyl group. So these are all amphipathic. Of course, these are your non-polar tails. So now that we understand how this structure, the head and two tails, are formed, they now have in response to that, they have the property that is now forming vesicular structures. So when you have these lipids that exist inside the body, if you remember the old commercials that say the body is made up of 2/3 water or 70% water, meaning the body forms an aqueous medium or a polar medium because maraming water. The polar heads interact fairly well with water and will associate with it. The hydrophobic tails want to run away from it and do not want to react with it. So free phospholipids or free sphingolipids in solution do not have thermodynamically stable configuration. But if you have enough of them, they are able to interact with each other because polar interacts with polar and non-polar will interact with non-polar. So they will form structures in a way that they will be able to thermodynamically satisfy solubility of both regions. This is also a spontaneous action. They will form it if they have enough, they exist in enough amounts and within close enough proximity. So the first example of your vesicular structure is the formation of micelles. So your phospholipids in aqueous medium are able to form micelles once they reach critical micelle concentration. They will form this having their hydrophilic heads facing outward towards the aqueous environment and their tails hidden inside to exclude water. So this is a cross-section of your micelle. The heads will be interacting outside, which is an aqueous region, and the tails are inside, hidden away from the aqueous region. Thing is, micelles can only grow so far because if too many mice, there will be space big enough for an aqueous solution to enter micelles if they become bigger. So to address that, they can instead form bilayers. This is the key structure when it comes to your biologic membranes, where they present with sheets forming where the hydrophilic heads, in a sense, sandwich both sets of hydrophobic tails. So this is a diagram of your lipid bilayer, where your hydrophobic tails are hidden or sequestered between your hydrophilic heads. When they're able to grow large enough, they can form vesicles where they now wrap around each other from end to end. So the outside is aqueous, the inside can be aqueous, and all the hydrophobic parts of your phospholipids are now secured inside. Question, yung bula po ba ay parang micelle? In a sense, yes, because the detergent or soap, any form of soap, is also like the structure of your phospholipid. The difference is it has only one tail rather than two. It has usually a salt, something like sodium or potassium as its polar side, and a long hydrophobic chain, or simply a a non-polar side that connects to dirt. Hence, soap is good at removing polar and non-polar dirt. Pero yes, parang bula, parang bubbles, kind of like muscles. Okay. So here, they are able to spontaneously form vesicles to protect and satisfy solubility of both the nonpolar and the polar sides. Okay. Next, the property of selective permeability makes it occurs because the lipid bilayer has very distinct hydrophobic zones that's impermeable to hydrophilic molecules. If something wants to pass through a lipid bilayer, it will first encounter a hydrophilic side, which may react with the hydrophilic molecule. But upon reaching the inner hydrophobic zone, will be stopped. So small non-reactive molecules maybe can diffuse through. Sterol like steroids, so sterol-like steroid. And hydrophobic molecules can easily cross the membrane. But non-lipid soluble molecules cannot pass through because they have to pass through a hydrophobic zone. In order for them to enter or exit the membrane, it has to be through membrane proteins that form channels, pores, or can serve as transporters. Next, the property of undergoing turnover. The membrane proteins, the membrane molecules that you have, so this bilayer in a sense, they're not permanent and they can be replaced over time. So the cell continually synthesizes more of these lipids and proteins in the intracellular side, specifically in the endoplasmic reticulum. And then these are transported out to replace old membrane molecules that are either damaged or utilized in cellular processes or in the formation of external vesicles. So that's the fourth property, undergoing turnover. The fifth is they can exist in asymmetry. They are asymmetric. So the distribution of lipids and proteins themselves are asymmetric, right? In two parts. There is an inside-outside asymmetry of proteins and lipids, and then there are also regional heterogeneities. First, the inside-outside asymmetry, where this accounts for the functional differences within the cell. And this asymmetry is also established as it was being made. You will see that sphingomyelin predominantly exists in the outer leaflet, while something like phosphatidylethanolamine consists more in the inner leaflet. The sugar moieties of glycosphingolipids and all glycoproteins typically protrude from the outward leaflet. And then this asymmetry is maintained by lipid transporters. So when we see that they're asymmetric, they can be moved from inside to outside or outside to inside, but they require transporters to do so. An example here is you see your green phospholipid in phosphatidylserine. Typically, these are found more on the inside, but when phosphatidylserine is on the outer layer, they can be transferred inward by a transporter called a flipase. Phosphatidylcholine, what is blue, typically on the inside can be brought out by a transporter called a flopase. So it's a flip-flop transport. It is easier to memorize. Flipase with an I moves cells from the outside going in. Flapase with an O moves cells out. These transporters are ATP requiring, so they are a form of active transport. Conversely, you have scramblases that shift two molecules around. This one does not use ATP, but it has to be active in the presence of calcium ions. So you see that these transporters, which are actual integral proteins, maintain the asymmetry of your membranes. Here's an example of glycocalyx, where the sugar moieties of your glycosphingolipids and glycoproteins will all protrude toward the outward inlet. There will be no carbohydrates facing inward. Okay. Next, property of asymmetry in regional heterogeneities. So as it says, in certain regions, there are increased concentrations of specific lipids and or proteins in the membrane. So these regions usually involve receptors, gap junctions, tight junctions, or synapses. Your cholesterol concentrations are also altered based on the specific needs of the cell and in response to external stimuli. So what do we mean by regional heterogeneities? For certain parts of a cell, there has to be there are functions of that cell that are performed by the proteins. The greater the number of proteins in that area, the greater the potential of the function, such as receptors or transporters or enzymes. So if that specific cell needs to produce, react to something, it can produce more receptors, hence making more receptors in a specific region of the cell. I will explain this a little bit later once we do a synthesis with the lipid bilayer. Okay. Next property is that they are dynamic. The positions of the membrane proteins and of the lipids, they're not locked in place. They are able to move around across the membrane plane. This fluidity of movement is definitely dependent on lipid and protein concentration, composition, and the temperature. They are able to do this due to non-covalent interactions among the lipids. So what do we mean by dynamic? They can be affected by external stimuli. So here, increasing temperature can drive hydrophobic tails to go from a gel-like phase to a fluid-like phase. So this is your non-polar heads, where upon the application of heat tend to move more fluidly. Example of this is like butter. Butter straight from the fridge, matigas. Once heat is applied, they become more fluid, which is similar because they are also hydrophobic chains. This dynamicity, or how dynamic they are, can be as a result also of your cis double bonds that cause the kinks in the tails and then leave more space for the fluid movement. So as you can see, is in an area that is full of saturated fatty acids, they are more compact, less able to move. With the kinks in the hydrocarbons, they allow more space in between your lipids. Hence, they are more compressible and they are more capable of movement because they are not as compact. Finally, cholesterol is a regulator of fluidity. It acts as a rigid structure. So because it is rigid, it is able to modulate fluidity in high temperatures where they cannot move cholesterol around because it is rigid. But in low temperatures, they cannot compress it because as the tails compress with each other. So it facilitates fluidity at low temperatures. Okay? Cholesterol decreases fluidity at high, but it retains fluidity at low temperatures. And of course, being in as dynamic, when they are able to move around, they can readily rotate, exchange, move laterally, forward and back within the same plate. The transverse movement across leaflets can occur spontaneously without your flapases, and uh, scramblaces, but at a rate that is extremely slow and non-observable. What is mentioning here is they can happen spontaneously. So we've discussed the 6 properties based on this of the biologic membranes based on the components that they have. So the components have properties when joined together exhibit more properties. So combine all together is what is known as the fluid mosaic model. Okay. It's the generally accepted model of a membrane structure, where the example is it's a sea of phospholipids, and then proteins are the icebergs. So again, culmination of the properties and its constituent molecules. Because it is a fluid mosaic model, the properties particular are the asymmetry of the planes, the fluidity of movement, selective permeability, flexibility, cost of that movement, and the ability to adjust to temperatures, and the ability to self-heal. Because self-healing meaning once an area is damaged or removed because of the spontaneous formation of vesicle structures and of undergoing turnover, they are able to heal. So here's an example of the fluid mosaic model. This is a sea of phospholipids, and these proteins are free to move around them. They are not locked in place. So all your proteins, your glycoproteins, your cholesterol embedded within. An example that I like to explain with the fluid mosaic model is this picture. Can anyone see this? Can everyone see this picture? This is an example of a crowded swimming pool in China. And typically what we love using to explain the fluid mosaic model. If a wave comes, all of them move up with the wave and move down. That is fluidity. The dynamicity is they can move between each other. Someone from the upper right corner can move down to the lower left corner with no disruption of no disruption of the membrane because they are tightly packed. They can be compressed. They can and they can also be laxed. What is they can allow more people in, and they can also prevent people from going out. Some form of permeability. Okay. So if you can imagine lang the situation, that is in a sense how it can look like in a more visual process. Okay. Next, we'll talk about the special domains of your uh, membrane. Just three to discuss. We have your lipid rafts, your caveolae, and tight junctions. So your lipid rafts, they are bilayer regions that are rich in cholesterol, sphingolipids, and then certain proteins. As we see cholesterol here and sphingolipids, which are tend to be more packed and less fluid or more regulated in their fluidity, they are more ordered. They're more tightly packed than the surrounding phospholipids. So in a sense, if a lipid raft is an entire family in that Chinese swimming pool that always has to move as a unit. Hindi pwedeng mawala yung isa because they are tightly bound by your cholesterol and your sphingolipids. They aim to segregate and concentrate certain proteins to facilitate their activity. So within that this particular lipid raft has a protein. If they need to increase the activity of that protein, lipid rafts can come together to form greater concentrations of that protein. These lipid rafts are involved in very specific processes. In this specific case, it's signal transduction. To the cell. They are specialized lipid rafts and their indentations is caused by the cholesterol binding protein calveolin 1. What do they do? So it's like a pocket for the cell membrane. It is potentially used for storage, for concentrating certain membrane proteins or certain receptors that when activated either release it towards the surface or vesiculize it towards the inside. So it's mainly used for endocytosis. I will explain that later. Signal transduction, like bringing it inside, or the regulation of proteins in lipid rafts. Last is tight junctions. These tight junctions are protein complexes that exist between epithelial cells. And they can contain proteins like occludin, claudins, and junction adhesion molecules. They are designed to maintain structure and to adjust permeability of epithelial cells. So these are cells that prevent diffusion of macromolecules between cells and in paracellular spaces. In the picture. Yeah. So example, this is the inner membrane of one epithelial cell, the inner membrane of another epithelial cell. These occludins, claudins, and other proteins form tighter bonds between the two cell membranes because this is the extracellular space. They do it to make the cells tighter and to restrict permeability. So imagine these rich in areas in the body where not even water should escape, such as the collecting ducts of your kidneys or for example, the inner anal region. Imagine if the inner anal region is permeable to water. Water can leak out from that area at any time. So these tight junctions are in greater concentrations in those specific regions where permeability has to be even tighter. Okay. Any questions before we move on to transport systems? Okay. If not, we can move on. So transport systems. We can understand that um, the membrane protects the cell. It prevents things from the inside going out, things from outside going in. And because the membrane is has selective permeability, it allows small inert molecules like gases in and out, no problem, and life of hydrophobic molecules in through the membrane. But how about other things? How do we acquire bigger molecules like sugars, like proteins, I mean, and amino acids, like hormones that form signals? Or how do we transport the new products of the cell going out? These are where transport systems come into play. So these are all protein complexes between epithelial cells. Yeah. So these are essential to cell function and communication, and allows movement of molecules through and against permeability. It allows the sending and receiving of signals and can either be passive or protein mediated. We have four general types of transport systems, from simple diffusion, protein mediated transport, and then for larger molecules, we have endocytosis and exocytosis. Simple diffusion is basically the passive flow of a solute from a higher to a lower concentration. Because it is doing that, it follows the electrochemical gradient, does not need to utilize proteins, but is limited in terms of temperature, is affected by temperature, concentration of the gradient across the membrane, and the solubility of the solute. Why this in particular? For temperature or thermal agitation, because the membrane is dynamic, the application of heat laxes the hydrophobic tails, making them more mobile, more able to move around, and more ability and decreasing these strict rigid areas where solutes can diffuse through. So increasing the temperature can increase um, the rate of diffusion. Consequently, of course, making it cold decreases the rate of diffusion. Concentration of the gradient across the membrane. Example is uh, it reaches towards an equilibrium. If there is a greater amount of solute from one side compared to the other, the movement across the gradient is stronger. An example is one side has 100 molecules, the other side has 0. There is a high rate of diffusion from the 100 to the 0. If you as compared to one side containing 60 and the other side containing 40. Because the concentration gradient is not as big, the rate of diffusion is slower. Finally, with regards to the solubility of the solute, the solute has to be soluble within the hydrophobic membrane. If it this is in terms of your solubility coefficient. Some molecules are more soluble in hydrophobic zones than others. The more hydrophobic it is, the greater the rate of diffusion. So what are affected by simple diffusion? Examples first are small and inert gases like carbon dioxide, oxygen gas, nitrogen gas, hydrogen sulfide. They don't really react to your hydrophilic or hydrophobic zones, even though they have polar zones. But because they are relatively small and don't dissociate, carbon dioxide doesn't break into carbon monoxide and oxygen, for example, they are inert and they don't react with the hydrophobic zones. So they are able to pass through. Small lipophilic molecules, lipophilic molecules or hydrophobic molecules are able to diffuse in, no problem. These are where your things like steroidal drugs come in. Water, to some extent, even though water is a polar molecule in a sense, is able to diffuse in because of its size. And because water joining with each other can move their polar zones inward to themselves. So what is not affected by diffusion? These are your inorganic ions and charged organic molecules that do not diffuse at a significant degree. Inorganic ions like sodium ions, calcium ions, chloride ions, and charged organic molecules. What are examples of charged organic molecules? Amino acids that have charge. Your glutamic acid, um, uh, your positive and negative amino acids. They are charged organic molecules. But they cannot diffuse into the cell. So if they cannot enter via diffusion, they have to be transported in via proteins. So this is your protein-mediated transport. Proteins that facilitate this form of transport are broken down into two types: your pores and channels and your transporters. So your pores and channels are aqueous. It's an aqueous pore in structure that has a controlling gate mechanism. Transporters, on the other hand, bind to your solute or your target molecule on the aqueous side before changing its conformation to transfer it through to the other side. It functions kind of like an enzyme. So the difference between the two is pores do not bind to the target molecule. It simply allows a passage in, kaya nga pore or channel. Well, transporters have to bind to the solute on one side before moving it through the membrane. And particularly, these proteins have a change in conformation as they do so. But once they release their target molecule, they reform back to their original shape or original conformation, and it does not change the target molecule. So parang enzyme talaga. So when we talk about pores and channels, it utilizes an aqueous pore that facilitates diffusion. It is fairly specific that specific pores only will allow certain things, and that specificity is based on their size and charge. Again, it does not bind to the target molecule, and it presents in alpha helical proteins and beta barrel proteins. So if you remember your lecture on proteins on secondary structures. Papakita na si alpha helix. And for beta barrels, these are sheets that form in round circular formations that, in a sense, form a large barrel. And since it's a form of facilitated diffusion, it still has to follow what the rates of diffusion. It goes down the concentration gradient. It does not use ATP. It is regulated by mechanisms that open or close the gates, and they can be inhibited by various compounds and other molecules. One example of your pores and channels in terms of your regulation is your voltage-gated ion channel. If you have discussed the action potential in physiology, this should not be a new thing to you. So for voltage-gated ion channels, they respond to changes in the transmembrane potential, or the difference of charge between the intracellular environment and the extracellular environment. These voltage-gated ion channels exist in excitable cells, for example, like neurons or muscles, where the depolarization of plasma membrane leads to the opening of sodium channels and then allows sodium ions in. Next is your ligand-gated ion channel, whereas its trigger is not a change in concentration, but by binding to a regulatory ligand. And that ligand produces an effect that changes the conformation of the channel, which may then open or close in response to the binding. An example of this is your nicotinic acetylcholine receptor. So the acetylcholine is your ligand, and the ion channel contains a receptor that is that has high affinity to acetylcholine or its agonist nicotine. So once nicotine or acetylcholine binds to the receptor side of the channel now changes its conformation to open. And because it now opens, it allows sodium ions in. I don't have a specific example, but I want you to understand the term of an agonist, which is something that mimics the ligand, that produces or that increases change. Like nicotine is an acetylcholine agonist because it promotes the acetylcholine receptor. Something that moves against the ligand is called an antagonist. Okay. So we're done with pores and channels. We'll talk about transporter proteins. So transporter proteins function like enzymes. They contain binding sites for specific molecules. Upon binding, they conform to move the molecule through itself and into the intracellular space. It then, upon releasing the molecule, returns to its original conformation, and then they do not change the conformation or structure of the target molecule that they have. They are proteins with their own specificity, defined kinetics, catalyzes a reaction. Instead of changing an enzyme to a product, it transports a molecule. Does not alter itself or its target, is unaffected by inhibitors. Hence, it functions in a sense like an enzyme. So here are samples of your transporter proteins. Or this is your sequence. First is recognition, from where an open transporter protein is bound to its target molecule. Part 2 is transport, where it changes its conformation, allowing the target molecule through itself and then through the membrane. Step 3 is to release the target molecule into the other side of the membrane, and then upon releasing, returns its conformational shape back to its original in recovery. So, parang enzyme pala. The transporter proteins also have classifications based on how they transport and what they transport. In this case, we have uniport, where uni1 transporting one molecule at a time. Symport is a form of co-transport where they move two molecules at a time, sometimes using the concentration gradient of one to drive the other. And the last form of co-transport is called antiport, where driving one molecule in is done through the gradient of driving one molecule out. So now that we've gone through the methods of protein-mediated transport, let's see its examples. First is facilitated diffusion. This is a protein-mediated form of transport that moves down the chemical gradient still. It is still diffusion, so it moves down the gradient and it cannot create a concentration gradient. It does not use ATP or energy, but these transports of proteins can utilize the downward movement of one concentration gradient to drive the translocation of another. This is called a secondary active transport that we can discuss later. The other form of protein-mediated transport is primary active transport. So for primary active transport, this requires the use of ATP. The transport protein, in a sense, binds with ATP and to become phosphorylated. ATP moves down to become ADP. The phosphorylation of the transport protein changes its conformation to facilitate the transfer of a molecule. And then once the molecule has moved in because of the transport, can become dephosphorylated to revert to its original conformation and receive another target molecule. So for active transport, the hydrolysis of ATP is used to drive movement. And then the catalyzation of the transport is against its concentration gradient. So for primary active transport, you can form and maintain an electrochemical gradient. An example, something that you may recall from the absorption of dietary proteins is your NAK ATPase for primary active transport. The function of NAK ATPase, or sodium potassium adenosine triphosphate ase, it exchanges three calcium sodium ions out when three sodium ions enter a transport protein or a transport NAK ATPase, with the phosphorylation from ATP, is able to accept three sodium ions. Upon dissociation of ADP, the phosphorylation of NAK ATPase changes its conformation to allow the three sodium ions out. While it's still phosphorylated, it is now in a conformation that allows binding of two potassium ions. And then once the phosphate, once it's dephosphorylated, when the phosphate group is dissociated, it reverts to its original position, moving the potassium ions in. So every cycle of NAK ATPase, this is basically the same ion, the same protein, a transporter protein in different phases. These are not different. These are not different. What do you call this? Transporter proteins, but rather the same one in different phases. Each cycle moves 3 ions out, two potassium ions in at the cost of ATP. So what does it do? In particular, it maintains the electrochemical gradient needed for the cell to be excited. You can see these types of other forms of primary active transport. You have your P-type, which includes your NAK ATPase. This table can be found in Harper. Your calcium regulated ATPase in your sarcoplasmic reticulum, your NAK ATPase in your plasma membrane. Your F-type proteins that you will see here, your mitochondrial ATP synthase used in oxidative phosphorylation. We won't go too deep on these types. I'm just showing you this table to show that there are multiple forms of ATP-driven active transport proteins. Your V-type or your vacuole type proteins, which include more present in your lysosomes and your synaptic vesicles. And then your ABC transporter or your ATP binding cassette transporters would be dealing with larger molecules. And going back earlier, an example for secondary active transport, it is an active transport that does not use ATP. The energy is driven through the downward movement of a concentration gradient of one molecule that is used to drive the translocation of another. You've probably seen this also in your digestion and absorption of proteins. The sodium glucose transporter, SGLT. Anyone taking or everyone familiar with drugs like your SGLT2 inhibitors? This is your protein. This is the transporter protein that is inhibited by that drug. So if you remember from your proteins lecture, sodium glucose transporter moves glucose from an area of low concentration to a high concentration. So this is against the gradient. But this is done through the facilitation of a high sodium gradient to a low sodium gradient environment. So you're able to pull glucose in against the gradient without using ATP because you are using the energy from the transport of sodium from a high concentration to a low concentration. So for secondary active transport, you are able to move against the gradient, hence of Symport because you use sodium. Your NAK ATPase, it's a form of antiport because you need to transfer out 3 sodium to enter 2 potassium ions in. So short summary, you have your passive transports. Your non-protein mediated transports is your simple diffusion. Your facilitated diffusion using pores and channels, like this one with the blue molecules, or your transporter proteins like your red molecules, are forms of facilitated diffusion. So they are protein protein mediated, but not active transport. For active transport, you are moving against the concentration gradient, the electrochemical gradient, and you require ATP. Okay, please familiarize and be able to differentiate the forms of transport. Here's a synthesis of a small synthesis of the of the transports that we have. We're looking at the proximal renal tubule lumen. So we're inside the kidneys. And we're looking at the action of SGLT, sodium glucose transporter. So from inside the kidneys, our urine has glucose and sodium in it. The proximal tubule cell would want to save as much of both as possible because they do not want to lose all the electrolytes and energy. So from the urine, the SGLT is the one that absorbs the transport sodium and glucose inside the cell through a matter of facilitated diffusion by secondary active transport against the concentration gradient of glucose, but through the concentration gradient of sodium. Your GLUT transporter is a uniport transporter that moves glucose from an area of high concentration to low concentration in the blood. So your glucose moves from the urine to the proximal tubule cell, then into the bloodstream by uniport GLUT transporter. And then to save your sodium, you have your NAK ATPase active transport, antiport active transport, moving your sodium back into the blood by exchanging potassium ions out. Okay. Next, so we're done with small molecules. How do we take in larger molecules? Endocytosis then is a process from cells to take up molecules too large for diffusion or too large for proteins. Using the process of vesicle formation within the membrane, you are able to take in those molecules by, in a sense, vaginating the membrane around the molecules that you want to take in. So these vesicles coming from the outside membrane form to become endocytotic vesicles, which can then fuse with lysozymes. So you have three types of endocytosis. You have your phagocytosis, or cell eating, phago, pinocytosis, or cell drinking, and receptor-mediated endocytosis. So here's an example. Your phagocytosis has specific targets, such as a solid particle here. The plasma membrane wraps around the target molecule and then end-to-end joins to form a vesicle. In this case, a vacuole. So for endocytosis, you are taking in a specific molecule. Not all cells are capable of phagocytosis, as we discussed in immunology naman. So we have things like your macrophages and your dendritic cells that are able to phagocytose because they have a specialized region in their plasma membrane known as pseudopods. In this case, you can see here the pseudopodium that are able to wrap around your solid particles. So phagocytosis is cell eating large molecules, but not all cells are capable of. Pinocytosis, or cell drinking, is taking the an aqueous amount of the outside outside environment going in that can trap other small molecules such as your sugars. All cells that have strong plasma membranes are capable of pinocytosis. And then finally is receptor-mediated endocytosis, where you have regions that are coated in receptors that are concentrated in a certain area and then moved in by the form of joining two ends of the vagination, hence moving in the receptor and the target molecule. Okay. So this, you see the red dots, these are examples. This is another diagram of caveolae, and these are caveolin proteins that assist in the formation of the vacuole, vesicle. So of course, if we need to move things in, we have to move things out also. This is the one that involves the clathrin. Okay, for exocytosis naman, you are releasing the vesicle from inside the cell. So we form vesicles from the inside of the endoplasmic reticulum that now are able to transport things out. Also, if not just cell components, but to move newly made phospholipids from the inside of the cell to the plasma membrane. So the endoplasmic reticulum forms vesicles that then fuse with the membrane, then releasing their contents outward. This exocytosis can also be regulated primarily by drugs and hormones. So here, an example here is your endoplasmic reticulum forms vesicles through its own internal membrane, plus the contents of that move through the aqueous inside and then bind with the outside or the main plasma membrane outside. If it can move proteins proteins and lipids that will bind to the new layer, or the vesicles can contain components made from inside the cell that has to be released outward. This form of exocytosis is a form of exocytosis done by pancreas beta islet cells that release circulatory vesicles full of insulin. The membrane binds with the plasma membrane, and then the insulin inside is released outward. Exocytosis can be modulated by hormones and other receptors that can use ligands, or can use steroids by entering through the plasma membrane itself. Okay. And last, short na lang to, is membrane receptors. So these are the membrane receptors are proteins that can be bound to specific molecules or react to specific stimuli to initiate a cellular response. So these receptors exist as soluble proteins and are bound to membranes that exist within the plasma membrane itself, and then can target the cytoplasm or the nucleus. The ligand is what you call the molecule that binds to the receptor. So when a ligand binds to the receptor then is altered by its tertiary or the quaternary structure, leading to conformational change, And that conformational change starts a cellular response. So the downstream effects of these cellular responses can be stimulation of growth and differentiation. An example is growth hormone. The up or down regulation of cell receptors. An example is LDL. Regulation of metabolic processes, that's insulin and glucagon is your best example for that. Stimulation of immunologic factors. These are receptors that you may be familiar to as your toll-like receptors, and the modulations of your nervous system. So this is just a small table from Devlin that shows the different kinds of receptors. So you have your G-coupled protein receptor, which an example of that is your glucagon and your acetylcholine receptors. Tyrosine kinase type of receptor like insulin and erythropoietin, and ion transport like glutamate and gamma aminobutyric acid. So again, this is just another table that shows different kinds of receptors, that there are multiple types of receptors for specific molecules, each with their own specific activity and or ligand. Yan. And actually, that's it. Does anyone have any questions? I understand na full day kayo, so I apologize na puro kwento and lecture lang. I hope I was able to spark some interest in you. So, nasend ko na rin ang